Assalamu alaikum, beta. I'm back with my next video on enzymes. I have collected all the questions from October-November session from the topic enzymes. And today I'll be doing the past paper questions of enzymes. Let's start. Let me share the screen with you. And then we will start with the questions without wasting time. Okay, here it is. I hope you can see the screen. These are all the questions from the topic enzymes. MCQs and structured as well, but right now I'm focusing on MCQs because you have your MCQ exam near. So let's start with the MCQs first. Okay, this is the first one. His first question says, a student was provided with two test tubes, one containing 10 centimeter cube of solution P, which we do not know what is this, and the other one containing 10 centimeter of solution Q. When these solutions were mixed together and left for 24 hours, the concentration of P decreased, but the concentration of C, its concentration decreased. Okay, दोनों को we mixed them, and when we mixed them, we found that after mixing, P decreased after mixing, and when we left it for 24 hours, we found it that P decreased, but Q remained the same. So the student wrote the following conclusions. P may break down over time. It can be correct because it might be a substrate which is broken down in the presence of this Q, which is the enzyme. Q may be a biological catalyst. Yes, that's what I have just said. Q can be an enzyme and P can be a substrate. So what will happen? The substrate will be converted into the product. So it will be decreased and Q will remain same because enzymes are needed in minute amount and enzymes because they are needed in minute amount. So they at the end of the reaction, they remain the same and even their concentration almost remain the same. So Q जो है वो सेम रहेगा और आपका जो P है वो decrease हो जाएगा. So they both are correct and P may be the substrate for Q, that is also correct. So from this, which conclusions can be supported by the information? So all of these three informations are correct. क्योंकि ये substrate हो सकता है, Q जो है वो enzyme हो सकता है. That's what they have written. P may be the substrate for Q in the last statement. So these are all correct. Which statements are correct for the lock and key hypothesis and the induced fit hypothesis? You know what is lock and key hypothesis, which was a conventional hypothesis that enzyme is behaving as a lock and substrate as a key, and if the shape of the key matches the shape of the lock, only then it can open the lock. That's what we know. So we call it lock and key hypothesis because it says that if the shape of the substrate is exactly same as that of the enzyme's active site, that only then it's going to fit into the active site. So that was lock and key hypothesis. But the latest theory is induced fit hypothesis, which says that both enzyme and substrate can slightly change their shapes in order to fit into each other, in order to, let's say, substrate fit into the enzyme. So in order to fit exactly, in order to fit properly, they both can change their shape slightly. So previously it was very a rigid thing, like a jigsaw puzzle, that if the shape is same, then they are going to fit into each other, otherwise no. But now it is a bit mild and induced fit. Yes, if the shapes slightly match of the substrate and the enzyme's active site, even then they can fit in, and in order to fit it properly, they both can change their shape slightly. So that's induced fit hypothesis, which is a bit mild, and that's the latest one. So, but they are saying ki which statements are correct for both of these. It should cater with both of these. So let's see. The substrate is the same shape as the active site. So again, that would be more suitable for lock and key hypothesis, but it's not for induced fit, where both can change their shape slightly in order to fit into each other exactly. So, and to let them react. So that's more with lock and key hypothesis, but it is not going with induced fit hypothesis. If we talk about this one, the substrate is held in place in the active site by temporary bonds. That's correct for both. That yes, the substrate is held in the active site by temporary bonds, which are the hydrogen bonds, and it fits and then it reacts. That is correct. The enzyme and sometimes the substrate change shape slightly as the substrate molecule enter the enzyme. That's correct for induced fit hypothesis, but it is not correct for the lock and key. Any lock and key doesn't say this can, yes, they can slightly change the shapes. So that's more with lock and key, sorry, induced fit, and this number one is more with lock and key, but this one is in between. Yes, substrate fits into the active site by making temporary bonds. So 2 is the correct option. Which one says 2? C. A student investigated the rate of enzyme activity with increasing substrate concentration. The experiment was repeated with the addition of a reversible, non-competitive. You know that inhibitors are reversible, like they do not bring in permanent changes. So if a non-competitive enzyme inhibitor is fit into the or is added to the reaction mixture, what is going to happen? And we all know that normally when the substrate concentration is increasing, okay, if the substrate concentration increases, then we get, and this is the rate of, on x-axis it's the substrate concentration, and this is the rate of reaction, which is an independent variable over here. And you know that independent variables are always plotted on y-axis and dependent are plotted on x-axis. Independent are plotted on x, I think I've said oppositely. Independent are plotted on x-axis, that's the substrate concentration over here, and rate of reaction is dependent on substrate concentration. More the substrate concentration, more is the rate of the reaction. So rate of reaction is the dependent variable over here, which is plotted on y-axis, and because it depends on the substrate concentration, and substrate concentration is the independent one, which is plotted on x-axis. So if we make a graph or plot a graph between substrate concentration and rate of reaction, we will find it that the rate of reaction continuously increases with increasing substrate concentration, but then it becomes constant. Yeah, I'm getting a rate of reaction becomes constant after a certain time, because at this point there are many other limiting factors which do not let the rate of reaction increase further. So it goes on at its maximum rate, which is the which is Vmax, but at constant. Like there's no further increase in the rate of the reaction. That's what we say. If we add a non-competitive inhibitor, enzyme inhibitor in the reaction mixture, the reaction will all be disturbed. Why will it be disturbed? Because if it's an enzyme and that's the active site, but this non-competitive doesn't attach to the active site, it attaches somewhere else other than the active site. Non-competitive inhibitor attaches somewhere else other than the active site of the enzyme, and because of its attachment, because what is enzyme? It's a globular protein. So when it is going to attach to this globular protein, there will be changes in this hydrogen, in the bonds within the enzyme molecule. As a result of these changes will force change in the shape of the active site as well. When the shape of the active site is temporarily changed, then the substrate won't be able to fit into it. If it's changed, if let's suppose it becomes this triangular or a square shaped or rectangular, then this substrate won't be able to fit into the active site of the enzyme because the shape is changed. So when this happens, then the reaction won't proceed further, and there is no further increase in the rate of the reaction, and the final rate, after the final rate of reaction, the rate of reaction reaches this point maximum. So without the inhibitor, that was the Vmax, but with the inhibitor, this is the Vmax, which is now low because the enzyme couldn't approach, and the rate of reaction could not approach the maximum rate of reaction because there is disturbance in the enzyme and the substrate, because the substrate is unable to attach to the enzyme now, or no more products are being formed. So that's why the rate of reaction has become constant again, and it is now more increasing because the substrate enzyme's active site is now not occupied by the substrate, so it's not being converted into the product. So no matter how much more concentration of substrate you are going to provide, but the rate of reaction will not further increase. So with a non-competitive inhibitor, this will be the graph, and without inhibitor, this will be the graph. So if we keep all this theory in our mind, and if we now check the graphs, which graph represents the result of the two experiments? You have seen that Vmax is now lower with the non-competitive inhibitor, Vmax lower. जबकि इसमें से तीन graph are those in which they are not showing that lowering of the Vmax. Instead, इसमें तो they have raised Vmax even from the normal case. They have increased Vmax so much. Here they are showing that Vmax reaches the same Vmax without the enzyme inhibitor, and here again they are showing the same thing. So these are all incorrect. This one is correct, where this upper graph is showing the that it is without inhibitor, and this one shows that it's with inhibitor, so the rate of reaction, which is the maximum rate of reaction, is now lower. Lower Vmax with non-competitive inhibitor. Okay, now the fourth one, which property of the tertiary structure of a globular protein enables it to catalyze a metabolic reaction? Catalyze कौन करता है? Enzymes करते हैं. तो let's see कि कौन सी ऐसी property है enzymes की, उनकी tertiary structure के globular protein की, which enable them to metabolize a reaction. It has a hydrophobic amino acid R. It has hydrophobic amino acid R groups on the outside. Absolutely wrong. Globular shape बनती तब है when hydrophobics reside inside, and all the hydrophilics surround the hydrophobics, and these surrounding hydrophilics are going to react with the external polar environment. So that's wrong, that hydrophobic means ki non-polar amino acids are on the outside. If they will be on the outside, then this globular enzyme won't be able to work anymore. So they are always trapped inside. These are non-polar, and these outer ones are polar amino acids actually. See, it will be denatured by high temperature. It is correct. But they are saying that on CSE property, which is enabling them to catalyze a metabolic reaction, which property is helping them in catalyzing the reaction? So denaturing at high temperature doesn't help. And that statement is incorrect. R groups of some amino acids form bonds with the substrate. That is correct. That from the enzyme, some of the amino acids have specific R groups which make bonds with the substrate. Okay, so these are the temporary hydrogen bonds made by the R groups of the active site with the substrate. That is correct. The three-dimensional shape depends on hydrogen bonding. No, not only hydrogen bonding, it depends on disulfide linkage. It depends on other properties as well, the ionic as well, hydrophobic as well. So it not only depends on hydrogen bonding. So the correct option is C. Astron investigated the effect of substrate concentration on the activity of an enzyme. The graph shows the results of this investigation. You can see substrate concentration is increasing, enzyme activity is increasing, and till this point, then some things will become limiting factor and the rate has become constant. But they are saying an increase in which factors could lead to a change in the activity of the enzyme at point X on the graph. इस point पे what is happening, in which factor could lead to change in the activity of the enzyme at point X on the graph? So an increase in which factors could lead to a change in the activity of the enzyme at point X on the graph? तो एक तो आपको साफ पता चल रहा है कि X पॉइंट पे सबस्ट्रेट की कॉन्सेंट्रेशन अभी बढ़ रही है, जिसकी वजह से रेट ऑफ रिएक्शन अभी बढ़ रहा है. सबस्ट्रेट की कॉन्सेंट्रेशन बढ़ाएंगे, इंक्रीज इन विच फैक्टर, सबस्ट्रेट की कॉन्सेंट्रेशन बढ़ाएंगे, तो इट कुड लीड टू चेंज इन एक्टिविटी ऑफ द एंजाइम. इट कुड इंक्रीज द रेट ऑफ एक्टिविटी ऑफ द एंजाइम. And if we change temperature at this point, if we increase temperature or if we change pH, then again there will be a change in the activity of the enzyme, because all these things are directly affecting the rate of the reaction. So all these options are correct, which are, if their concentrations are being changed, they're going to directly affect the rate of the reaction of the enzyme. So all are correct. Michaelis-Menten constant Km is the substrate concentration at which an enzyme works at half its maximum rate. We all know that Km is when this is half of Vmax. The enzyme has what is correct for when the Km is low? Now the Km is low, it means that low Km means that this enzyme whose Km is low, Michaelis Menten constant is low, it has a very strong affinity, high affinity for the substrate. That is why the reaction, half of the Vmax has approached very quickly, and the rest of the reaction will also proceed very quickly because the enzyme has a strong affinity for its substrate. So low affinity वाले तो हम गलत कर देंगे. High affinity for the substrate, and the quicker the reaction will proceed to its maximum rate. That's C. इसमें वो कह रहे हैं and the slower the reaction will proceed. गलत. जब उसकी affinity strong है तो the reaction will also proceed quickly. Some molecules fluoresce when illuminated with ultraviolet light. Changes in the structure or shape of such molecules can change the intensity of the fluorescence. The enzyme tryptophan synthase fluoresces bluish green because of a phosphate group associated with the active site. The enzyme catalyzes the reaction as shown. So this is the enzyme which is actually bluish green because of the phosphate group in it. But this can be changed when there is change in the concentration of phosphate groups of the enzyme. So this enzyme is catalyzing the reaction between serine and indole, and tryptophan is being formed in the presence of the tryptophan synthetase enzyme. Adding serine to enzyme increases the intensity of fluorescence. अब वो result भी it's being shared कि when we added serine to we realized that the intensity of fluorescence increases. But when indole is also added, the bluish green fluorescence decrease in intensity. तो वो क्यों डिक्रीज हो जाएगी? What may be concluded from these observations? Serine and indole attach to the active site of the enzyme. Yes, they do attach to the active site of the enzyme, and of course, the changes will happen in the active site. Then in enzyme, serine complex is formed. Yes, complex between the substrate and the enzyme is being formed. And enzyme, serine, and indole complex is formed. That is also correct. All of these three can be the reasons of decrease in the bluish green fluorescence. Yeast contains the enzyme catalyst which catalyzes the breakdown of hydrogen peroxide. You can say कि enzyme है catalyst जो yeast में, and that's breaking down hydrogen peroxide into water and oxygen. And we recorded the results after every 30 seconds for 2 minutes. यानी 30 सेकंड पे रिकॉर्ड किया, 60 पे किया, और उसके बाद आपने 1 मिनट पे किया. इस तरह से आपने उसकी रेंजेस बना ली. When you record it, to realize that with time the total volume of oxygen, ये जो इसी reaction के end पे बन रही है वो oxygen है. Total volume of oxygen. It was in the beginning 157, then it became 251, 283, and 285. So you can see that there's a huge gap between 151 and 251. Yani after half minute to 1 minute, there's a huge gap and increase in concentration of oxygen. But in the second minute, from 130 and then total to after 2 minutes, there is the volume of oxygen is decreasing. So in the first step, it was so high, but then it is decreasing. So what explains the pattern of the data? The rate of reaction increases as more oxygen. The rate of reaction, यहाँ पे rate of reaction तो वो बता ही नहीं रहा, वो तो volume of oxygen बता रहा है. तो we will not consider them correct. We can think about these two. The volume of oxygen released decreases as the enzyme begin to denature. If enzyme starts it doesn't enzyme doesn't denature on low or normal temperatures. The volume of oxygen release decreases as the enzyme begin to denature. So this doesn't show denaturation. There is increase in the concentration in the volume of oxygen. So it says that the volume of oxygen release decreases as the enzyme begin to denature. That's wrong. The volume of oxygen release decreases as more substrate is converted into product. ठीक है, the volume of oxygen release decrease क्यों हुई है यहाँ पर, क्यों decrease होती जा रही है, गैप कम आता जा रहा है 283 से 285 only, जबकि यहाँ कितना गैप है 30 से 60 मिनट पे. So you can see the volume of oxygen release decrease हो रही है, अब उसकी एक ratio जो है वो decrease हो रही है as more substrate is converted into product, because it's being converted into product. So we need to add more substrate as well at this point. The matrix maintain constant Km is a measure of affinity of an enzyme for its substrate. ठीक है? So the higher the affinity, the lower the Km. Correct. Reaction will proceed quickly. The lower the affinity, the slower the reaction will be. That's also correct. At Km, half the active site of the enzyme are occupied by the substrate. That's also correct because Km is half of Vmax. So at Km, half the active site of the enzyme are occupied by the substrate. That's correct. All three are correct. Now we have structured questions, so we will discuss the structured question later on. We have discussed all the MCQs of enzymes. I hope you understood those and you can tell me about those things which are not clear, which are difficult for you in all the topics. I will one by one be focusing on those topics as well. Thank you very much. Allah Hafiz. We'll see you with the next video, inshallah, on mitotic cell cycle and DNA and protein synthesis, inshallah. Hope we'll see you again.